Unit 3 is the big unit. This is the one with all of the brain parts, the brain regions, the association areas, the lobes, the hemispheres, the parts of the neuron, all of the scans, neurotransmitters, hormones, nervous systems. This is a huge chapter. It's a chapter that's a big part of the curriculum, but it's also one that is some of the most common questions that are asked as far as review is concerned. So let's jump into chapter 3 or unit 3, which is the biological basis of behavior, and it's every type of brain function. All right, so to start out with the neurological function of the brain, we have to start out at the most basic unit of the brain's tissue, which obviously is a neuron. Several neurons make up a nerve, and there are millions of nerves inside various nervous systems in the body. So to start at the most basic type of tissue, let's talk about the parts of a neuron. Now, the neuron that you see here is actually a central nervous system neuron. It's a brain neuron. There are different types of neurons, and they all have the same parts and the same action potential order, which is just a neural impulse, I should say. But they have different shapes. Some sensory neurons or motor neurons can be this exact shape, kind of look like a palm tree, but they have much longer axon cables. In fact, some of them stretch the entire length of, say, the femur, or the entire length of parts of the arm, and they can be very similar to this, but in different sizes. There are other types of brain neurons or central nervous system neurons that have different shapes than this. They have the cell body in the middle with axon cables coming out of both directions that feed in to the dendrites. They have the terminals coming off of the cell body in the middle. There's various different types and shapes just depending on the function of the neuron. But for the most part, this neuron is the type that you would see in a diagram on the AP exam. So a neuron consists of a nucleus, which is the cell body itself. That's where all of the action potential happens. And basically what happens is charges, electromagnetic charges, or I should say electrochemical charges, activate in the dendrites. The dendrites are the branching extensions of the cell body. That term comes from the Latin word dendra for branch, so think of like the branches of a tree. If a tree were vertical and something were to fall from the sky, it would land or be caught in the branches, in the dendrites. So the dendrites are where the messages actually create a static change. And once you get depolarization, the cell body basically creates a neural impulse, which is called an action potential, and that opens up they're not pores, that opens up the pathways in the axon cable because the neuron is trying to polarize itself again. So you get negative ions and positive ions and ions that are being released from the neuron itself, which causes the electrical pulse to travel down the axon and out of the sending end of the neuron, which is the terminal, the branch terminals that come off of the axon. So the whole entire order of a neural impulse is from dendrite to cell body, down the axon cable, and out the terminals, or the terminal branches, which are the extensions or the nodes that are at the end of the axon cable. Think like a battery has a positive terminal at one end, that's the end that sticks out, and it has a negative terminal for receiving a current at the other end, and a neuron is no different. To encase the axon and insulate it, you have a specific type of glial cell, which is known as myelin, and the myelin kind of creates a sheath around the axon terminal itself. And when that myelin breaks down, you get various disorders. There are certain disorders that cause the degeneration of myelinated tissue, and that can cause anything from multiple sclerosis if this neuron was part of a body system, a somatic system, or like a motor system, or if it was in the brain, in the central nervous system, it would be something more like dementia or Alzheimer's if the myelin were to break down. So neurons communicate with each other in a chain which is known as a pathway. And how neurons communicate is through neurotransmitters. So to understand the process of action potential, the neurotransmitters travel around in the gray matter of the brain. They land on receptor sites of the dendrites, and that causes a change in the polarization of the cell body, and that's how you get a thought, or that's how you get a message from one part of the brain to the other along a pathway. And as long as that action potential is strong enough to keep reactivating the next neuron in the chain, then that neural process will continue, whether it's a thought or a memory or motor function, whatever the case may be. So to look at the actual chemicals that remain in your brain, we'll get into the chemicals here in detail, but there are certain things to keep in mind, and one is the idea of the blood-brain barrier. So the glial cells that feed the neurons and myelinate the axons also act as the nutrition in the brain. And there are different types of glial cells, just like there are different types of neurons. And there are certain types of glial cells that only concentrate in the brain. And those are known as endothelial cells. And those types of cells actually, what they do is they insulate and protect, and they supply nutrition to neurons that are in the brain. But the other benefit to those endothelial cells is they actually encase the neurons in the brain so successfully that they prevent neurotransmitters from escaping into the bloodstream, and they actually prevent toxins that are in the bloodstream from escaping into the synapses, which are in between the neurons. And so that's known as the blood-brain barrier, which protects neurotransmitters in the brain from escaping into the bloodstream, but it also protects toxins in the bloodstream from affecting the brain in a negative way. So to understand neurotransmitters and understand, you have to kind of understand or visualize that process of a neural impulse, that molecules that are floating around in the space of the brain, they bounce around in those synapses in between neurons, and when they land on receptor sites, they can concentrate and they can create an action potential. So to look at some of the major neurotransmitters and their functions, you have things like ACH, dopamine, serotonin, norepinephrine, GABA, and glutamate. To look at this from a more emotional standpoint, the most significant neurotransmitters that you would be asked about, say on an FRQ, would be something that has to do with a specific function. So to put this into a bigger picture and hopefully help with comprehension, let's look at something like the reward pathway. So you have emotional responses in the brain. And that causes emotional responses in the body. And a lot of people are very confused with certain neurotransmitters like dopamine. What does dopamine do compared to what is serotonin? What does dopamine do in comparison to, say, something like oxytocin? How does cortisol come into play? And so there's other chemicals that you don't see on this chart that are very, very, very important in understanding emotional responses in the brain. So for instance, if we look at the reward pathway, which is what's known as dopaminergic, meaning that it processes and metabolizes dopamine. So dopamine is a very complicated molecule. It has various functions depending on which part of the brain or which pathway in the brain is activating. So to make it simple, let's think about it this way. You have positive molecules in your brain, and you have, let's say, negative molecules in your brain. And typically when we say positive and negative, we're talking about adding or subtracting. But in this case, I mean positive as in these are good moods, good emotional responses, versus negative, which are bad emotional responses. So dopamine exists in a part of the brain that's known as the reward pathway. It's actually called the mesolimbic pathway is what it's called. But dopamine is one of these molecules in your brain that actually motivates you to do something. And the reason why dopamine is necessary, if you look at how these molecules affect each other, dopamine, serotonin, and oxytocin are the positive emotional chemicals that affect the brain. So a good mood, a motivated mood, or the opposite of anxiety, the opposite of stress, the opposite of depression is going to be a combination of dopamine, serotonin, and oxytocin. So you look at oxytocin, which is not on this chart. Oxytocin is social trust between humans, really between animals. If you're looking at the oxytocin releases in primates, it's very similar to that of humans, even in things like lobsters and rats. Crustaceans, they actually have serotonin pathways in their brain similar to ours as humans. But serotonin is the mood molecule. That's what the more serotonin you have, the more manic that you become. but it also regulates a positive balance in your mood. So if someone's suffering from depression, it's going to be a depletion of serotonin. Their serotonin levels are too low. But there are other things that can cause your serotonin levels to dip. Saying that you're depressed doesn't necessarily mean that you have chronic depression. If somebody has a serotonin insufficiency, meaning their brain does not produce or does not process or metabolize enough serotonin, then that would be a psychological disorder. That would be a neurological disorder, and you would need medication. You would need an antagonist like a SSRI, which would be like a selective serotonin reuptake inhibitor. So, but there are normal times in our day and in our life that you can have a reduction in serotonin because your brain is reacting to stimuli. So serotonin, dopamine, oxytocin, those are things that affect and change our mood. Where oxytocin is trust, it's bonding, it's a positive experience that you have between two beings. In this case, we'll say humans. So when you have a positive experience with another human, it's going to release oxytocin in the brain. That's how you bond with them. That's how you learn to trust them. And that's kind of an evolutionary adaptation. Think in the case of primates. If a primate is going to allow another primate to groom their fur, then they have to allow another animal to get close enough to them to harm them and potentially kill them. So that requires a lot of trust. And so oxytocin is the trust hormone, and sometimes it's called the cuddle hormone. It is the neurotransmitter that builds a bond. And certainly it can be romantic. You know, I've heard it explained by one psychologist that oxytocin is certainly released during sex, but we all know that you can have sex without trust and without bond, and you may not have that same oxytocin release. And so it's not so much the act of sex that would build a bond between two people. It would be the bond between two people that would increase the oxytocin during a sexual act. So oxytocin is also the chemical that's released during contractions at childbirth, and that's to allow that labor to continue and the birthing process to progress. But it also has an evolutionary function which allows mothers to bond with their newborn babies. So dopamine, serotonin, and oxytocin all kind of work together as positive emotions. And when you experience something like negative emotional responses, depression, you could have a lowered amount of serotonin, meaning that your mood is too low. So conventional wisdom would say, okay, well, let's raise our serotonin. Well, how do we raise our serotonin? Well, if serotonin affects your mood, that's something that you have to increase in the brain. You have to stimulate the brain to increase the amount of serotonin that it has. So if you're suffering from depression, your brain is not releasing enough serotonin. Well, when you look at the studies of other animals, primates, and certainly even humans, obviously, you get serotonin releases that happen from accomplishments. In fact, there are animals in the animal kingdom that only release serotonin when they, say, win a fight. So if two animals, in an alpha dominance desire fight one another and one of them wins, there's a serotonin increase there. There's a different posture that animal has. It increases their mood. It increases their sense of accomplishment. And so serotonin is kind of a very fickle thing. It can drop and it can increase. And so there's activities that we can participate in that increase our serotonin and make us feel better. There's also activities we can participate in that increase our oxytocin, which help us bond with other people, which also increases our serotonin. But it's important to remember that all of these molecules are metabolized, meaning that they're broken down. So if you get an increase in serotonin or if you get an increase in dopamine, that's not going to last forever. That's something that lasts for 15 to 20 minutes, and then it's going to get metabolized. So the brain, in an effort for homeostasis, is constantly regulating and evaluating. That's what the hypothalamus does, evaluating which chemicals are in ready supply and which ones need to be increased. So to apply that to everyday life, if you suffer from anxiety or if you're having an anxious attack or a panic attack, basically what that is a depletion of serotonin, it's a depletion of oxytocin, it's probably a depletion of dopamine, but the other thing that you have is it's an increase in a negative chemical, and that's a chemical that you don't see on this chart, but that is the stress hormone, which is called cortisol. Cortisol is what happens, what's released in the brain when we perceive a threat. So now that we don't have threats to our physical safety from predators or environmental things in our civilized society now, or our advanced society, I should say, cortisol is still a chemical that's active in the brain. And so what we release cortisol, or a stress response, for is actually not a physical threat. It's a threat. It's not a threat to our safety. It's going to be something like test anxiety, or it's going to be something like response to environmental stimuli, like say this COVID-19. It's going to release the stress hormones in the brain. And so to deplete that anxiety, or to burn off that anxiety, that stress is going to be metabolized also. Cortisol does not last forever in the brain, just like dopamine doesn't last forever in the brain, or serotonin doesn't last forever in the brain. Cortisol has a time limit, but in order to eliminate that, you have to increase the serotonin in the brain, you have to increase the dopamine in the brain, and even doing things like increasing the oxytocin in the brain, those positive chemicals can actually counteract the negative chemicals. So it's hard to isolate one neurotransmitter and just say, okay, this is what dopamine does, right? This is what serotonin does, because they all kind of work together. For instance, dopamine being one of the most complicated ones. If you look at how the mesolimbic pathway works, or what mostly is referred to as the reward pathway, it has different parts, right? You get things like the nucleus accumbens and the ventral tegmental area, the VTA. All this is part of the reward pathway in the brain, the screenshot that I stole from Khan Academy here. And basically what happens with dopamine is this. We talked early on in the year when we were covering chapter 3, we talked about this theory that's known as the reward prediction error as a way to explain dopamine. So the reason why dopamine has to exist, and the reason why we have a reward pathway, is because we need a motivating chemical to get us up off of the ground and to go seek and do. We need to eat, we need to fill our bellies, we need to find mates, we need to fight off predators. And so this is an ancient chemical response in the brain. It's important to notice where in the brain it's taking place. This is the limbic system of the brain. This is the middle of the brain. This is what's known as our mammal brain. So if you get under that prefrontal cortex, which is labeled there on the screen, that's the gray matter of the brain. That's in the frontal lobe. That's what separates the human attributes, we'll say, from other even advanced mammals. Those are things like impulse control, decision making, sense of humor, advanced memory processing, and logical mathematical processing, making predictions about the future, evaluating judgments, and things like that. It's a very uniquely human traits. But underneath the gray matter of the brain, underneath the cortex of the brain, once you start getting into the white matter and the middle parts of the brain, you have a brain that's known as the mammal brain, the limbic system, which you share with other mammals. And they have the same responses. So for instance, to look at it a different way, the dopamine reward pathway in the brain exists in the mammal brain. So other mammals also have that. So to look at it realistically, all animals make decisions. But most animals make decisions based on the chemical drives in their brain, whereas humans can also use that prefrontal cortex and make decisions that are separate or different from our emotional responses. So when we think that we might make decisions solely based on knowledge or past experience, or a hunch, in reality, that's only one way to explain a very complex system in the brain. We might be making our decisions based on a fear response, or based on motivation from dopamine, or a reward that we get from an activity that was successful. So understanding dopamine, the easiest way to explain it, which is oversimplifying it, is that it is a motivating molecule. It's the molecule that encourages us to go and seek something. But how dopamine works in the mesolimbic pathway, the reward pathway, is that you have to constantly increase the dopamine to get that satisfaction. So whenever we are motivated to participate in a behavior, and that behavior has a desired outcome, there's going to be also dopamine activation in that VTA, in that ventral tegmental area. And basically what that is those are the dopamine releasing neurons, the pathway of dopamine neurons that are in the limbic system of the brain. So that VTA is where that dopamine is being produced and where it's being released. And that's all part of this reward pathway, this mesolimbic pathway. So what happens is that notice that the hippocampus, which is what forms long-term memories, explicit memories. So that's a limbic function also. And most people don't think of memories as being an emotional response. But the reality is this: the purpose that for having memories at all is to make judgments based on past outcomes. So if we participate in an activity and we get a positive outcome from that, like you're hungry and you go get a sandwich and the sandwich is delicious, then basically what's happening in that reward pathway is the memory triggers in the hippocampus are getting involved, so that you can file that away as a successful action or behavior, so that when you're hungry again, it's easy for your brain to go back and find a behavior that led to a desired outcome. And so that's a very complex process, I know, but it's way too simple to just say that dopamine is a motivator, because without dopamine we wouldn't get up and seek, we wouldn't go find food, we wouldn't chase after a potential mate. Dopamine is involved in excitement, it's involved in sexual arousal and desire, it's involved in hunger, it's involved in a lot of different processes. And if you notice the amygdala there, that is fear and aggression, which is attached to the hippocampus, and that's obviously a very strong motivating intense emotion, a positive emotion, an additional emotion, we'll say, that is very strongly tied into memory. So all of these things come together, this limbic emotion that dopamine stimulates, this limbic system, are definitely for the purposes of survival, but what it does is it makes it easy for us to make judgment based on past experiences. So when you have a negative experience and your brain remembers that, it's the hippocampus that's storing those memories of events, and it's going to fire again and tell you, remember last time this was bad. We don't want to do that again. And so that allows you to make judgments based on past experiences. And that's why dopamine is such an important part of the neurotransmitters. So to go back again, let's just take a look at them. You have dopamine, which influences movement, learning, attention, and emotion, but that's a very boiled down way to explain it. Dopamine is the motivating molecule. It is the reward molecule, but it's a very complicated molecule that's involved in various functions. And again, the dopaminergic pathways in the brain, where dopamine is produced and dopamine is created, dopamine also is metabolized. So dopamine doesn't last forever, and that's what happens in things like drug addiction. So when you get a dopamine increase from a positive outcome, then essentially your brain is filing that away. Well, the next time that you go and ingest that substance, you're not going to get the same level of satisfaction, because the brain is expecting a certain amount of novelty. And that's the downside to the dopamine reward pathways. You get drug addictions, and not only are you addicted to a substance, it doesn't even have to be a substance, it can be an activity, it can be gambling, pornography, or something like that. You are motivated and stimulated, and you get a positive outcome, and your brain files that away as a positive experience. But dopamine works on excess, on more. And so when you seek and you find a positive outcome, your brain is desiring an increase in dopamine, and so it's not enough to have the same level of dopamine as last time that you participated. It wants the increased amount. It wants the excitement and the anticipation as well. So serotonin again is mood. Norepinephrine would be the part of the neurotransmitter in the brain that stimulates fight or flight. In fact, it's partly responsible for cortisol and anxiety. And so when you look at anti-anxiety medication, some of them are SNRIs, and that's selective norepinephrine reuptake inhibitors. And what they do is they prevent norepinephrine from increasing in massive amounts and regulating the amount of norepinephrine in the brain for a healthy level of mood control. To understand GABA, you have to understand glutamate. And glutamate, what glutamate does is glutamate is the molecule in the brain that stimulates neurotransmitters to fire. So let's say that it facilitates neurotransmission. Or put another way, glutamate is the chemical that allows your brain to work. It's the chemical that allows your brain to communicate. And glutamate is kind of a nutritional element that goes to the neurons themselves. So you need glutamate in order for your brain to work. The problem is that there are synthetic forms of glutamate in our diet, and a lot of times we eat foods that have bound gluten in them. And some people who have gluten intolerance, or they get migraines from gluten, the reason for that is because the type of gluten that we eat, which is a preservative in foods, the brain reads those gluten molecules as if they were glutamate. And so what happens is that you have a lot of overstimulation of the brain. You have a lot of overcommunication of too many neurons, and that can cause headaches, it can cause migraines, it can cause anxiety, it can cause us to ruminate and stress too much because our brain is just overstimulated. It's working too much because there's too much of that glutamate in the brain. So the inhibitory neurotransmitter that counteracts glutamate is GABA. And GABA basically is the counteractor to glutamate, which causes some of these neurons to calm down and stop firing so much. ACH is what's enables muscle control, control of muscular function, somatic muscular function. And so when you have too much ACH, essentially you can get your muscles seizing up, you can get too much overstimulation, tremors and things like that. When you don't have enough ACH, you can get things like a complete lack of muscular control or even paralysis. All right, so moving into synthetic materials. These are other vocab words that often get confused, and those are agonists versus antagonists. So to understand what agonists and antagonists are, we had to have our previous discussion. We had to have our previous explanation. Agonists and antagonists are the term that's used for synthetic materials that end up in the bloodstream. So anything that you take orally, that you drink, that you inject, that you accidentally breathe in that's in the air, anything that's in the food that you eat, any foreign substance that gets into the bloodstream and activates the brain is either an agonist or an antagonist. So most people think of drugs, but it's other things like diseases. You have meningitis and infections and things like that. There are toxins that are in the air or in the food that you eat. We're talking about things that affect the brain, things that are what's called psychoactive. They activate the mind. And of all of the substances that you will encounter, only 4% of them are psychoactive. So you can eat foods that have toxins in them, but they may not affect the brain because they can't get through the blood-brain barrier. So these are the things that are psychoactive, but they have to be synthetic. They're things that come from outside of your brain's normal chemistry. So it's easiest to explain agonists and antagonists by talking about drugs. So if you have a headache, you might take medication that's going to block a pain signal. If it blocks a pain signal, it's going to be an antagonist. If it's something that mimics a brain's chemical reaction or a molecule in the brain, it's going to be known as an agonist. So agonists are drugs that occupy receptors and they activate them. Antagonists, or the anti, are drugs that occupy receptor cells but do not activate them. They block them. So essentially, if you look at the chart there, it says agonist alone is so chemically close to a molecule that your brain produces that it actually mimics the effects of that molecule. So think opioids. What happens when you take something like a prescription painkiller that's an opioid, a narcotic, essentially is that's such a molecule that's chemically almost identical to an endorphin. And endorphins are pain-blocking, euphoric feelings that your brain releases when you need to fight pain, when you need to block pain. And sometimes you do things like you go for a run and you get a runner's high, and endorphins are released in the brain and you can't feel pain anymore. And in fact, you feel great. You have this euphoric feeling. You don't feel aches and pains, etc. So that's what endorphins do. Well, there are synthetic forms of endorphins on the market, and those are narcotic drugs. Those are known as agonists. So drugs that occupy receptor sites that the brain confuses for endorphins. So when you take an opioid and your brain thinks that you have too many endorphins, then it will stop producing them because it can't metabolize the endorphins fast enough, and essentially you get an addiction. You get an endorphin depletion. Something that's an antagonist is essentially close enough to fit into the receptor site in between the two neurons, but it's not chemically pure enough to activate the neuron to fire. So that would be more like something like marijuana as opposed to an opioid. What it does is it blocks cannabinoids, right? It blocks the ability to release dopamine in certain parts of the brain. So that's going to have an effect by not allowing a chemical that your brain produces, instead of like a painkiller, which has an effect on your brain by allowing or causing, let's call it exciting versus inhibiting. So if it excites the brain's actions, it's an agonist, and if it's a foreign substance that blocks a brain process, it's known as an antagonist. So there are two different major nervous systems in the body. You have the nerve, and they don't communicate entirely directly to one another. So you have three types of neuron in your body. You have interneurons in the brain and spine. You have motor neurons, which are for movement, and you have sensory neurons, which bring messages into the brain. So essentially, what happens is the brain and spine have to translate signals from the body, and the brain has to send signals to the body. So motor messages are outbound messages from the brain, from the central nervous system to the peripheral nervous system. And sensory messages, like pain, for instance, or touch, temperature changes, those are messages that come from the neurons in the skin cells, and they go to the brain, and those messages are interpreted in the spine. They're interpreted in interneurons. So to understand the different nervous pathways in the body, you have to understand that there are three different types of neuron. So the peripheral nervous system is going to be everything that excludes the brain and spine. So very literally, the center of your body, if you were to cut it symmetrically in half, like so on the screen, the central nervous system is the brain, the brain's tissue, and the spinal tissue. Everything else is the peripheral nervous system. And the peripheral nervous system has two different functionalities. You have the autonomic soft tissue organs and glands, the endocrine glands, and you have the somatic or motor function of the body. So think S for skeletal, S for somatic. So soma means body. So this is physical movement and things like pain signals. The autonomic controls are going to be things that the hypothalamus and the pituitary gland do. So if it's soft tissue, organs, hormones. If they mention hormones, it's an autonomic function. And the autonomic function is broken down into two categories. You have glands and organs in your body that ramp you up for arousal. Those are known as sympathetic nerves. And you have counteracting glands and hormones in your body that are released to calm you back down. And those are parasympathetic nerves. So basically, if your brain perceives a threat, you may get something like epinephrine, which is adrenaline. That's a sympathetic molecule which increases the body's alertness. So there's only so much of that you can tolerate because you're burning energy and you're metabolizing calories, and your brain just can't maintain a high level of alertness all the time. And so what happens is, once the perceived threat is gone, a counteracting molecule is released in your system, which will calm you back down. So you have epinephrine, and then you have a counteractor to epinephrine. So you have things that will increase, say, your hunger, and then you have things that will decrease your hunger, things that will ramp you up, which are sympathetic, and things that will calm you down, which are parasympathetic molecules. So these are hormones. Hormones are in the bloodstream, whereas neurotransmitters are in the brain, in the central nervous system. Hormones are peripheral, and neurotransmitters are central. Here's a closer look at the different glands and organs in the endocrine system. These are going to be parts of your peripheral nervous system that either ramp your system back up or calm your system down. So examples of sympathetic nervous arousal would be dilating of the pupils, an increase in the heartbeat. Digestion is inhibited in the stomach. Basically what happens is glucose is released by the liver, and what happens here is that your body is preparing to deal with a potential threat. So for us, that may not be something like you're being attacked by a predatory animal. It might be something like you're about to go sit and take the AP exam and do FRQs, and you're really stressed out. So the sympathetic nervous system is to counteract threats, fight or flight. But a lot of times in FRQs, they want to know a physiological symptom of sympathetic nervous arousal. So if you get a question that says, what is a physiological example of sympathetic nervous arousal, don't say fight or flight. Say an increase in the heart rate, or a dilation of the pupils, or say something like relaxation of the bladder. Right? Those are examples of physiological responses. If they say a physiological response for parasympathetic or calming you would say the pupils contract again, the heartbeat slows back down to normal, digestion is stimulated again, the gallbladder is stimulated, the bladder contracts back again instead of relaxing, and you get normal blood flow. So basically what happens is sympathetic arousal in response to a threat, and you get parasympathetic calming so that you can go back down to normal. So think of parasympathetic like a parachute coming back down to homeostasis, which is what happens every time that you have an increase in certain chemicals in your bloodstream, which are hormones. You have to come back down to normal. And that's the constant struggle of the hypothalamus, is to keep you normal. A simple reflex just involves the spine. There are motor functions that the spine can send instructions for, like tissue damage and pain, without by bypassing the brain, we'll say. The endocrine system we discussed, that's the soft tissue and organs in the body that create hormones and secrete them. So an important distinguishment is the hypothalamus itself is part of the limbic system. It is not really a gland because it does not create and secrete hormones. The pituitary gland does. The pituitary gland, as you can see, that green figure there, is outside of the blood-brain barrier. It actually kind of hangs down from the limbic system. That's where the hormones are created and released, secreted into the bloodstream. The hypothalamus is really just deciding how much of what chemical that you need, and it's giving instructions to the pituitary gland. The pituitary gland is the factory where those molecules or those hormones are actually created and puts them into the bloodstream. So you have things like the thyroid and the parathyroids, the adrenal gland, the pancreas, and of course in the sexual response cycle you have the sex organs for reproduction. Part of sympathetic nervous arousal would be sexual arousal, and then the resolution after climax would be the return to homeostasis with blood flow and decreased heart rate and decreased stimulation. A lot of people ask questions about the scans. So all the scans are listed here. You can see the various types of scans, of ways that researchers can check the brain, and depending on what it is that they need to find, some of these are more invasive than others, some of them are more expensive than others, some of them are more problematic and difficult to use than others. So essentially the easiest and least invasive way to check brain activity is an electroencephalogram. So think gram equals wave. So if you're checking your heart rate just to see when it spikes and how often it does, then you're going to do an EKG, which is an echocardiogram. But if you're going to put electrodes on the brain, on the head, cephala is head, so if you're going to put electrodes on the brain, on the head, then you're going to see brain wave activity. Now it does tell us, as you can see in the image there with the little girl, that your brain is active and how active it is. It does not tell us what part of the brain is active, or what part of the brain is breaking down glucose, what part of the brain is heating up, or what's going on in various parts of the brain. It's not specific, but it is brain wave activity where those electrodes are. So if you put electrodes on Broca's area and ask a child to speak, you're going to see brain wave spikes from that particular electrode that's in that part of the brain. So you can see brain activity in parts of the head, which is very helpful, but you can't necessarily see specifics. When you look at the PET scan, by comparison, the PET scan is going to put radioactive glucose into your bloodstream, and as that blood circulates through the brain, you're going to see the metabolism of the sugar. So essentially, if your brain white matter runs on glucose, which is how the nutrients that your brain needs to function, and your brain uses about 25% of the calories that you ingest. That's how much energy it takes just to function with your brain. You can see that as the brain is active in particular tasks, it lights up like a radar, where you can see that sugar is being metabolized. An MRI, when you look at the arrow there, looks at you can see it's a black and white image of soft tissue. It's a little bit more detailed than an X-ray. Because it's using just a different technology. It's using magnets. When you look at a functional MRI or an fMRI, basically what you get is you get changes, and those changes can be highlighted on the screen. So what happens is you get those MRI images, and when something changes, the machine that you're using is going to highlight those changes or highlight the change or the contrast on the screen. It's going to kind of show you still images of how things have changed in the brain during a particular activity. A CT scan, which is the big tube. Some of you might have gone through a CT scan for a concussion, or you've definitely seen it done in movies and things like that. A CT scan is basically layers of X-ray images, and all it will show you is damage to the brain, or it will show you the tissue itself. If you're looking for skull fractures or bleeding, swelling, tumors, which is what we see here in the CT scan, especially in image A, you can see that large tumor and what looks like the left temporal lobe of that patient. So those are things that would show up on a CT scan. Parts of the brain. So we'll get into regions of the brain and parts of the brain. We've really discussed the limbic system at detail, so we're not going to spend the same amount of time in detail on the limbic system, but you can see all of it there. It's kind of overwhelming to think about the various parts of the brain and what they do. But basically, each of the lobes of the brain have specific functions. Right, so the occipital lobe is where the visual cortex is. That's where sight data is going to be processed. The parietal lobe is going to be things like recognition and understanding of various things. It's your brain making sense of the world based on the sensory data that it gets. So that's where you're going to get like the somatosensory cortex or the feature detectors. You're going to get the frontal lobe, which is a significant amount of your brain's surface, your brain's cortex tissue, which is uniquely advanced mammals, especially humans. And so and the temporal lobe, which you can't see here on this chart because it kind of points to the middle, is things like the auditory cortex and the limbic system itself. Other parts of specific parts of the brain, we'll get into things like Wernicke's area, Broca's area, visual memory, processing, the sensory cortex, and the motor cortex. So starting with the brain stem, it's broken down here for you in vocab terms. The brain stem consists of the medulla, the pons, the cerebellum, which you can see is called the little brain there. You have the reticular formation that runs through the cerebellum, and the thalamus, which sits on top of the pons. So the medulla is controlling your breathing and heart rate. It's at the base of the brainstem. It actually narrows down into the spinal column itself. The pons is kind of that pinkish color that's labeled there in the chart. It sits on top of the medulla and helps to coordinate movements. The reticular formation runs throughout the inner parts of the medulla, and that is what alerts you or wakes you up. And that's also how they can put you under. By blocking action in the reticular formation. So if you get a pain message because they're cutting open your knee in a surgery, that message is being sent from the tissue, but the reticular formation is not communicating with the brain to become alert, to become conscious and aware. So the reticular formation is what keeps you awake and alert, but when you get spinal damage high enough, if it damages the reticular formation, you can go into a coma, or some cases even die. So the thalamus at the top of the brainstem is the sensory switchboard, and that's the part that takes all of the sensory information coming in from the body and routes it to the various location in the brain. Right? So the cerebellum is hand-eye coordination. It's going to be voluntary movements and things like that. The limbic system we talked about, the amygdala is fear and aggression. The limbic system is the middle brain. Right? To go backwards, if we look at what is the oldest parts of the brain, the dinosaur brain, or what's often referred to as the reptilian brain, these are things that you share with reptiles. That's the reptile brain, the older brain structures. This is the mammal brain. So more advanced animals, more evolved animals, we'll say, have emotional responses like we do. So you have things like the amygdala, which is fear and aggression. You have the hypothalamus, which is what checks the chemicals in the bloodstream to maintain what chemicals need to be released. So the hypothalamus is all of those drives that you have in drive reduction theory. It's going to be hunger, it's going to be thirst, it's going to be sexual arousal, things like that. Body temperature, and it controls the pituitary gland as well. You have the hippocampus. The hippocampus is explicit memories, long-term memories. We'll talk specifically about that when we get to Chapter 7 or Unit 7. Of course, the phrase that we used was you would not forget seeing a hippo on campus, and that's a mnemonic for the hippocampus, meaning memory, long-term memory. The cortex of the brain is the gray matter. The white matter is what runs through the middle. That's think white tissue, like fatty tissue. Those are glial cells. The gray matter is where the actual cell bodies are. That's the surface of your brain. So on the surface of your brain, we are very, very much different than our animal counterparts. Once you get past the cortex of the brain and into the middle parts of the brain and the white matter of the brain, we are identical to other animals like primates and things like that. So the surface of the brain, the cortex of the brain, is an important part of the brain to understand because it's what makes us as humans significantly different, especially in the cerebral cortex, which is in the frontal lobe, the prefrontal cortex. So you look at the cerebral cortex, which is essentially just the cover, the surface, the M&M shell, if you will, of the brain itself. The frontal lobe is the lobe that's behind your head. The parietal lobe is at the top, above the temporal lobe. The temporal lobe itself is in where your temples would be if you take two index fingers and put them on the sides of your head. When you have headaches and pressure in the temple, that's the temporal lobe. That's the auditory cortex, where you process sound and speech comprehension in the left temporal lobe, Wernicke's area. And then you have the occipital lobe, which is in the back of your head. For some reason, sight is processed in the back of your head for things that you see in front of your skull. That's where the visual cortex is and where sight data is processed. There are two cortexes of the brain that are important to remember, and people often mix them up. You have the motor cortex and the somatosensory cortex. The motor cortex is that strip of tissue in each of your brain's hemispheres that controls movement. So your left motor cortex is going to control the movement in the right half of your body. So if you get a stroke in a certain part of the corpus callosum, that's going to prevent those motor messages from getting across to the other hemisphere, and you would have paralysis or lack of physical control in that half of your body. If you get a stroke closer to the frontal lobe, when it comes to that corpus callosum, if the corpus callosum tears, in the frontal parts of your brain, you wouldn't have paralysis necessarily, but you would have what's called aphasia, and that's essentially your speech problem because of where the messages cannot cross into the opposite hemisphere. So the motor cortex of the brain is what controls the physical movements for the opposite half of the body. The sensory cortex, the somatosensory cortex, is where the incoming messages are processed from the brain. So if someone touches your arm, it's going to be felt, quote unquote, in the somatosensory cortex. If you move your arm, that instruction is going to come from the motor cortex. So motor is movement, and you can use that context clue on an FRQ. If they ask anything with motor neurons, motor cortex, that's going to be controlling physical movements. Like the FRQ we wrote, Ashley needs her motor neurons to move her hands and to move her feet to drive a car. I added language areas here just to help you out. A lot of people confuse Broca's area with Wernicke's area and the angular gyrus. These are specific parts of the brain that are all interconnected for the not only comprehension and processing of speech, but also the creation of speech as well. So I mentioned aphasia in the previous slide. Aphasia is what happens when language is impaired from a stroke. Usually, it's a language impairment or inability because the corpus callosum is damaged, where the speech processing parts of the brain are supposed to travel. Wernicke's area is the reception of structured language, the comprehension of it. So think of it this way: Wernicke's area is necessary to understand and process communication. Even if someone was writing something down or using hand gestures or saying something out loud, however it is they're communicating with you, it's Wernicke's area of the brain that's going to process that this is language and not just random things that I'm viewing. There's a pattern here. That's not just a squiggly line segment, that's an S. So that's what Wernicke's area of the brain is going to do. Broca's area is what's going to allow you to physically form the words with your mouth. It's the expression of language, the control of muscular movement of the mouth, the tongue, the jaw, and the cheek so that you can form letters. So if you have damage to Broca's area, you can still understand and comprehend and probably even write and draw what it is you were trying to communicate, but you'd have a difficult time trying to express it. So aphasia is sometimes known as Broca's aphasia. It's not a comprehension problem, it's an expression problem. The angular gyrus is visual language info that's connected together that would allow you to read something and comprehend it on a page while you were saying it out loud at the same time. So it's going to be the interconnector or the go-between between Wernicke's area and Broca's area. In split brain research, again, the corpus callosum is that band of tissue that connects the left hemisphere with the right hemisphere, and sometimes if you have a stroke, you can see the corpus callosum in the bottom right of the screen there. It's highlighted in the limbic system. It's a big thick band of tissue where axons cross from the right to left hemisphere. So if you sever that, or if it gets cut or torn for whatever reason, you get an absence of communication from the left hemisphere to the right. So your right hand control is in your left hemisphere, and your left hand control is in your right hemisphere, and you may not be able to send those signals if the corpus callosum were severed or damaged. I added this for hemispheric specialization because a lot of times when you look at charts that are online, and I actually had a difficult time finding one that was fairly accurate, they give very specific examples of what the right half of the brain does compared to the left half of the brain, and it's really not as simple as saying, well, math is in your left hemisphere and art is in your right hemisphere. The reason why those exist is because the right hemisphere and the left hemisphere actually create a picture together. The left hemisphere depends on the right, and the right hemisphere depends on the left. So basically what you have are small details versus big picture items. And if you didn't have access to your right hemisphere, you would not be able to see the greater picture in things. You would only understand tiny, minute details. You would not be able to understand it as a bigger part. You would not be able to use a gut intuition. You would not be able to use creativity. You would not be able to assume and presume, really, based on past experiences and understanding the things around you. But if you didn't have access to your left hemisphere, you couldn't analyze. You couldn't use algorithmic processes. You couldn't use logic. You would only have big picture things. You couldn't understand small details. You couldn't create language. And you certainly couldn't understand number use or patterns and things like that. Our concept of time or awareness of time. Again, time is a human understanding. Sometimes I heard something that was it's silly, but it was profound, and people were talking about the life cycle of certain insects, like a fly. Well, it lives 3 days, and you think, well, does a fly comprehend how short his life is? Well, all life is just relative. All time is relative. So the fly does not have the ability in its brain, its little fly brain, to understand the concept of time. Even the idea that one day ends and another begins based on whether or not it's nighttime or the sun, is orbiting or the earth is orbiting, those are all human constructs. We've defined our life in time. We've defined our life in years, and we've started to comprehend our mortality because eventually time passes and we grow closer to the end. So there's some kind of horrifying philosophical thoughts that humans wrestle with that animals don't have to. And that's part of the explanation of the difference between, say, a dog's brain and a human's brain. I mean, it's one thing for us to struggle over the fact that a dog's emotional responses and decisions are different than ours because they don't have that prefrontal cortex tissue, but it's another thing to understand hemispheric specialization, to say that humans are actually capable of comprehending these things. In fact, our stress and cortisol and our panic and anxiety come from our understanding of things around us. Little children, they don't have anxiety over pandemics or their mortality and the fact that someday they'll get old and sick and die. It's not that they don't choose to think about those things, it's that their developing brain can't comprehend these existential crises that we can in a more developed brain. And certainly the same is true for animals. Your dog doesn't comprehend that he's going through his midlife dog crisis and that he only has half of his life left and that he's stressing about the fact that he hasn't accomplished the things that he wanted to accomplish by the time he reached the age of 7, and all of these things that are just silly to even imagine with an animal. It's because of the very advanced left hemispheric specialization of the prefrontal cortex. So it's kind of an interesting thing to think about, that humans, ironically, are beyond environmental threats like animals are. We don't have predators, but yet we can ruminate to the point of a panic attack and take medication because our anxiety is so bad, because we're stressed out about things that might happen in the future. It's really actually kind of fascinating to think about. So when we get into the parts of the brain, again, there's just things that you have to kind of know. The difference between the right and left hemisphere, what the 4 lobes of each hemisphere are, and I mentioned specialization in the last slide. There's also a term that you might see in an FRQ, and that's the lateralization of the brain. The lateralization of the brain refers to what is similar. Specialization of the hemispheres is what is this part of the left hemisphere do compared to this part of the right. So an example of hemispheric specialization would be the fact that the left frontal lobe has Broca's area in it. So that's a specialized part that's just in the left. The right frontal lobe does not have a speech processing center. So that would be a specialized part. Lateralization means exactly what it sounds like that it should mean. It is that of the parts of the brain that are symmetrical and identical. So the somatosensory cortex, for instance, is lateralized because you have the same shape and tissue band for sensory information in your left hemisphere, and it's identical and symmetrical for the opposite half of your body in the right hemisphere. So hemispheric specialization is what's different about this part of the brain, whereas hemispheric lateralization is what's the same about these symmetrical parts of the brain. It's just another look at the specialized parts of the brain. Now we get into things like heredity and heritability. Another commonly asked question is the difference between fraternal twins and identical twins, so I added that in here as well. Remember, fraternal twins are separate fertilized eggs. So one sperm cell that fertilizes one egg is a human. You get 23 chromosomes from the mom's tissue, and you get 23 chromosomes from dad's sperm tissue, or cell. So basically what happens is that's the chemical recipe for the human genome, is 23 pairs of chromosomes. But as you know, because you probably have siblings, that even siblings that are fraternal are very different in a lot of ways. So even the same two parents that mix their cellular information together multiple times, they're going to create different people with different traits. Even the difference between fraternal twins, one being male and the other being female. So fraternal twins are no more genetically similar than other siblings. They just, both of their eggs were fertilized at the same time, and they share a uterus together. So they obviously have a lot of connection in their environmental things because they're the same age and they go through life stages at the same time, and so they're probably close, but they don't have the genetic similarity that identical twins do. Identical twins are actually the same fertilized egg, which splits, or let's use the word, is cloned, we'll say, into two separate functioning people. It's really kind of a fascinating thing. Identical twins is a single fertilized egg, which splits into two feti. The fraternal twins are just two eggs that are fertilized by two cells at the same time. And that is genetic, whether or not you are more likely to have twins. This is also from chapter 11. Heritability is mentioned in chapter 11, but this is a chart that is an interesting example of heritability. Heritability is how likely is a trait the result of genetics. So if you're 6'4" and your brother is 5'4", is that because your genetics are that different? Or is that because that he had some stunt in his growth? Is that because you're 27 and he's 17? How likely is it genetic? So even people who have the same genetic parents have genetic dissimilarities, differences. So basically, when we can create the same environmental factors and we get differences in people, then we can point to that as being heritable. So for example, when you look at the plants, you have a uniform nutrient solution, which means they all have the same amount of nutrients, they all have the same access to light, they all get the same amount of water, yet some of the plants are significantly taller than other ones. Well, if the environmental factors are the same, then the difference in size in the plants has to be because of the genetic makeup of the plants. So in this case, the height of a plant is going to be heritable. You can't give a pine tree the same nurturing environmental factors as a dandelion and say, wow, I must have watered that pine tree so much more than I watered the dandelion because look at how much taller the pine tree is. Well, it's a different species of plant, obviously. So basically, the difference between the groups being genetic or the difference between the groups being environmental. If it's the same species of plant and you're getting the same environmental factors, there's something genetically different about the other plants that are taller. Now, as the diagram also shows, what if you have the same species of plant that gets different environmental factors? So if you get more sunlight on some of the plants, or more water concentration on some of the plants, or more nutrients and fertilizer on some of the plants, then they may have grown taller just because of an environmental difference, right? So for instance, when you look at the difference, the example it uses in the book is in places like North Korea where there's stunted nutrition. Well, that's not a heritable trait. The North Korean children who are significantly shorter than their South Korean genetic brethren, it's the result of those height differences is not because of genetic differences between the North Korean children and the South Korean children. They are shorter because of environmental factors like a lack of nutrition. So that's heritability. Evolutionary psychology, we mentioned in the chapter 2 review. That's this idea of competition for survival leads to the advancement of a species. And then basically as the species adapts to its environment, it develops adaptive traits. Natural selection, Darwinism, survival, and reproduction. So that's the end of chapter 3. That's a long amount of information, I know, but there are significant amounts of questions that typically come from chapter 3. Stay tuned for the next video where we will cover Unit 4, which is sensation and perception.